Good morning. My name is Liz Horn, and I'm the Director of Genetic Alliance Biobank, and I want to thank you all for attending the webinar today. Today we're going to be talking about making your organization's registry and/or biobank a reality. And we have two fantastic presenters from two different organizations, Kim McCleary from the CFIDS Association of America and Megan O'Boyle from the Phelan-McDermott Syndrome Foundation. But before we get started, I wanted to take a few seconds to go over some housekeeping things. First of all, everyone on the call is muted, so we can't hear you. So in order to ask your questions, you'll have to type them into the question panel of your webinar box. And I also want to let you know that the presentation slides can be downloaded from our website, geneticalliance.org/webinar. And the recording will be available next week, and we will send you an email to let you know when it's recorded and up on the website. So before I turn it over to Kim, I wanted to tell you a little bit about why we thought this kind of webinar was really important. We know that registries and biobanks are really important tools for patient advocacy organizations. We also know that it can seem like a daunting task to set one up. And there are a lot of pieces, both technical but also organizational. There's a logistical side of this. And what we're trying to do at Genetic Alliance is to provide some forums to discuss this information. So I'm very, very excited that our panelists are here to share their stories. We will open it up for questions at the end. We hope to have a really robust question and answer session. And I will also be sharing some of the tools we've created because we know this can be challenging, and we want to help you as you start to think about this. So Kim, I'd love to turn it over to you. Kim is the President and CEO of CFIDS Association of America, and they just started the biobank this year in 2010.
Right. So just a little bit of background. I'm not sure how familiar our participants today might be with chronic fatigue syndrome, and because it is not a rare genetic disorder in the way that many of the partners of the Genetic Alliance are, I just thought I'd take a couple of minutes to go quickly through some of the research challenges that have been barriers to more discovery about CFS that led us to create a biobank as a solution and a research accelerator. So our organization has been around since 1987. We are the largest CFS patient advocacy research support organization in the country and actually in the world. And we have since day one in 1987 always been very interested and directly supportive of research studies. So being involved in research was not a new thing for us when we started thinking about a biobank, but it was a very different strategy from funding investigator-initiated grants, which had been sort of our traditional way of supporting CFS research. So, and again, just some points about chronic fatigue syndrome so that you understand the sorts of challenges that we were facing and why a biobank made so much sense for us at the time. Well, there are at least 1 million Americans affected by CFS, so again, this is a pretty common condition. It has a very variable phenotype in that many patients, at least a quarter of them, are completely disabled, and some of them homebound and bedbound, while others function pretty well and are able to hold down jobs but may have to eliminate all the other activities in their lives. So you have a pretty wide range of severity that has to be accounted for. There have been at least 7, and there's a brand new one that just came out last month, definitions used for research to select subjects for CFS research, which has created problems in terms of the heterogeneity of the patient cohorts that have been tested and examined and studied for the last 25 years. There's no single diagnostic test that can be used to define patients, and while there's a characteristic symptom pattern, as I said, it can present itself in quite a range of severity, duration, and also just the symptom patterns that are most prominent within an individual may change over time and may be different from person to person. It's 4 times as common in women, so it follows a pattern of many autoimmune diseases. There have been many infectious agents suspected and linked to CFS over time. It typically affects adults, but children and teens get it usually around the time of adolescence. And it occurs in all ethnic and racial groups and at all income and education levels. And there are also family clusters and workplace clusters that suggest both genetic links and common exposures. So we had sort of, all of these challenges kind of going in. If I can get this to advance. I spoke a little bit about the heterogeneity, and I put this slide up. It's my colleague Suzanne Vernon's, I think, favorite slide, just to demonstrate that this is a very heterogeneous group of people. There may be many different causes and triggers that lead to the clinical symptom complex known as chronic fatigue syndrome. So Suzanne Vernon, who is our research director and came from CDC in 17 years at CDC before joining our staff, is a systems biologist, but she also has an epidemiology background. So one of her priorities in coming to give our research program more internal capacity was to find a way to help sort this heterogeneous group of individuals into more homogeneous groups of smaller individuals that could then be characterized by biomarkers, molecular subtyping, so that clinical and pharmacogenomic interventions could be more targeted and hopefully more effective. So this is kind of just by way of background of what we were trying to do in our research program circa 2008. The other challenge that we had kind of going in is that basically everywhere any researcher has looked in the body for abnormalities in CFS, they have found them. And these are just kind of mapped out here. So we have all these researchers taking different parts, kind of like the story of the men feeling the different parts of the elephant. And they found sometimes remarkable and sometimes subtle disturbances in all of these different body systems, which creates kind of a problem on this end of where do you target therapy and where do you continue to target research to be as efficient with your investments and in creating kind of the next important discovery. So one of the things that we did at the beginning of 2009 was to create a CFS research network, so the beginnings of one. We funded 6 investigators. This is Suzanne, our research director. We funded 6 research projects in 3 of them are actually in New York. 1 is in Wisconsin, 1 is in Canada, the other's in Utah, and we linked them through a research network and enforced as a condition of our funding that they would use some common instruments to collect information about patients, some common samples that they would all collect on all of the subjects that they studied, and we would try to start making sort of this common platform and lexicon that all of these 6 investigators could then use when their results were ready to compare from site to site as a step forward toward all of those challenges that had become so apparent after the first quarter century of research on this condition. What we found was it was really hard to standardize. There were differences among the approaches they were each taking that led them to want to study one type of patient versus another. Dr. Meadow here, who's looking at the autonomic nervous system because those abnormalities are more prevalent in young people, had a really young age range of patients he wanted to study, whereas Kathy Light in Utah, the clinician that she was working with, had access to more of a older and more female patient base, and that's who she had to study. So it was hard to get them to even agree on certain parameters about the subjects that they would jointly look at. And he needed one type of blood sample and she needed a different type of blood sample using different tubes, so it was hard to get them to kind of agree on something that they could both use and that would work for both of the different hypotheses. There were also issues of each IRB having to approve any changes that collectively we wanted to approve, taking different lengths of time to get through University of Alberta or Marshfield or NYU to make sure that everybody could use those same elements. So what we decided after sort of moving through that process and getting a little way into it was that we needed to be more central to create this platform and to make sure that we had investigators who were both linked to one another and whose patients could be linked through a common platform as a means to move some of the information and some of the early science from bench to bedside. And we became very convinced that a biobank was a way to do that. So the biorepository helped us address these particular challenges and certainly many more, and I know Megan is going to share some of the ones that their organization faced. But it gave us the ability to draw participants across geographic boundaries. So now, rather than being confined to where those 6 bubbles were on that map, we can enroll participants from really anywhere across the country, and we're also enrolling people from Canada and other countries. We were able to begin with a consistent entry criteria for subject selection, and we kind of took the best of the 7 different research definitions for CFS, and based on some studies that have been done by other groups, we were able to sort of pinpoint what criteria we thought would make for the best cohort without limiting the number of people who would meet that criteria too much. And it would also give us the ability that if 2 investigators wanted to study different types of patients, say a group who had a very infectious type onset versus a group who had trauma as an onset, we would likely have enough individuals who met those sort of sub-criteria that we could help that investigator out. The biorepository allows us to collect consistent data on all the individuals. We don't have to rely on what different tools might be in use at a different academic center or clinical center. Because we have the opportunity in the way that Genetic Alliance has arranged its IRB structure, we have the ability to go back to participants over time, and we will be able to follow both the clinical course of the illness and also collect samples at different points in the future, both to replenish samples that might get used in the course of research studies, but also so that we can maybe have an annual sampling point so that we have some longitudinal data as another feature of our asset. One of the issues I think that plagues all sort of clinically based research is the cost that investigators have to invest in enrolling subjects. So with this biobank now, we can help bring that cost down because we're doing a lot of the work that they would have to do ahead of time, and we have the sample cohorts all ready for them to be able to use once they've applied for access to the information and gone through the IRBs at their own locations. We also have the ability, because of the foresight of Genetic Alliance, to be able to customize the sample collection to reach, to support different hypotheses about the illness. So if somebody wants to look at the blood, they have, we can use different tubes. We can get plasma. We can get whole blood. It can be processed according to their customized needs. But if somebody else wants to look at genetic material from a local tissue swab, we can do that. And I know there are other groups that are collecting other types of tissues for their research. So we see this as something that's definitely scalable and is a growth opportunity as the research advances. We also wanted a way to take one of those sites on our map of 6 people, the findings at those sites, and to validate them across different sites. And with a biobank, we have that opportunity as long as they're blood-based markers, or patients can be recruited in for special, like brain scans and things from different locations if that research moves on and needs to be expanded to a different population. And I think one of the most important things that we discovered in conceiving of the biobank and then doing some due diligence as far as what other opportunities were out there as a support structure for the biobank is the importance of the institutional review board approval. And this is totally necessary for the protection of people involved in the research, but it's a huge hurdle for people in academia to cross. Some of the investigators that we funded in the past are able to get their IRBs squared away within a couple of months, and for others it may take 6 to 8 months. So having the patient selection and enrollment piece of that taken out of the equation and centralized to our biobank has really been a tremendous advantage in terms of people being able to move forward on bench science where the samples are already collected and then sent to them. Also, in terms of the informed consent part, making our participants feel comfortable that their information and their samples will be protected and that their privacy and confidentiality issues are being handled in an appropriate way has been very important to our population as far as gaining support and participation for the biobank. It has been somewhat challenging, and it's, I think, a product of the current environment in CFS research. There's been a recent discovery of a new human retrovirus being linked to CFS called XMRV, and there is no FDA-approved test for XMRV. So many people who have inquired about our biobank are looking for a way to get tested for XMRV, and there has been some resistance to the idea that they, as participants in our biobank, will not get personalized information about test results that come out of research that's supported through the biobank. But ultimately, I think the people who are motivated by the desire to participate in something bigger than themselves and to contribute to medical understanding and scientific advances have enrolled quite readily and understand that there are some trade-offs, but none of them in terms of the risk of their personal information being used for other commercial-type purposes. So in short, all of those challenges that we had, we really found that the biorepository and specifically the Genetic Alliance biobank infrastructure has been a tremendous advantage to us in accelerating research. And there are some other biorepositories of tissue, and specifically blood for CFS, but what sort of unique advantage we have had in serving the CFS research field is that our biobank is scalable, and I talked about how we were able to sort of select a set of criteria that allow us to be broad in terms of capturing a wide cross-section of the CFS patient population, while at the same time the tools on the back end allow us to be able to be quite narrow in selecting patients for specific studies. And I think I would put that in terms for a broader audience of being scalable, that whether the condition is rare or common and whether it often has comorbidities or is on a scale of other conditions, the biobank really can be customized and scaled to reflect whatever the particular population is that's being studied. And then I've just listed some of the advantages from an operational standpoint about the infrastructure that we have, that somebody's freezer full of tubes that may have been collected over time by different people doesn't necessarily provide. And again, that's the institutional review board and the informed consent pieces. We know that everybody who goes in understands what their sample has been collected for and how it might be used. The web-based collection of standardized clinical information gives you a huge sort of data mining tool on the back end. The blood and tissue sample collection can be processed according to standard procedures and also customized for different hypotheses. Inventorying and tracking samples is not as simple as knowing when the blood gets drawn and when it gets delivered. Sometimes those samples are split into different portions, and some of them are used and some of them are kept. And it is quite a complicated process, especially when you start acquiring a large number of participants. And then just also the data management and data querying tools are really strong advantages of this system. And the security and access control. I think everybody's read horror stories about different information systems being hacked or compromised in some way, and it was very important to us that those measures for security and access control were well addressed by the vendors that we looked at. And then just the tremendous knowledge base and expertise that you're engaged with through Genetic Alliance. Liz alone is a huge resource, and all of the other people who've been down this path in creating biobanks has just been a great added benefit to being part of this network. So, and I just wanted to give a quick sort of summary of how quickly, actually, we were able to mobilize and get this started. It's been less than a year, although the concept of doing a biorepository and registry is something that both Suzanne Vernon, our scientific director, and I had been talking about for quite a while, even before she joined our staff. So it's been an interest in something that at first the Centers for Disease Control and Prevention had said that they were going to take on, and then did so in a very limited way, and that we ultimately, by early 2009, with the experience of trying to sort of, centralize our research network, realized that we were going to need to add another layer of effort to make this happen. And Suzanne met with some of the people from Genetic Alliance at a meeting in September 2009, and we did kind of an expedited due diligence process of looking and evaluating different options that was ultimately concluded in November, and then our board approved our partnership with Genetic Alliance in December. We got our application in January, and the IRB came through in early March, and we made our announcement to the community on March 29th. And that was for targeted enrollment of a narrower, defined group of patients that were going to be recruited through 4 clinical centers just to get us started. And we actually began our first study collaboration around that same time. So we were even initially collecting samples for a very specific study. Our first sample came in April, and then we announced a general enrollment, which is as much like a registry as it is a biobank, on June 8th to expand out to the number of people that were interested in participating but didn't meet those initial sort of refined criteria. And now, as of this week, we have 450 individuals, which includes some healthy controls, enrolled in the biobank. So for less than a year's work, we're very happy with how this has moved forward. And we very much see the biobank as a catalyst for translational research. For us, it has been a very cost-effective way to provide a robust resource for research. It has created opportunities for cooperative research. We have the one study that we started with in June, and we are in various stages of discussion with 4 other outside organizations who are interested in collecting, in receiving some of our samples and clinical information. So that has, already been very productive. And I should say that we are making our samples available on a per-cost basis, so that has helped us support financially the expenditures that we've made to build this resource. And, the stakeholder community, the patients and the advocates, just really have been tremendously receptive to the idea of participating directly in research without having to travel to a specialized clinic across the country or without being screened out because they didn't have access to a certain provider or a certain academic institution. So I think it's been a very positive thing in terms of the perception of the association throughout the stakeholder community. And then finally, just as an asset, we've created this asset as a hub for quality biologic samples and standardized clinical information, which is sort of a proof of concept. The very early slides I showed with sort of the chaos of the research in CFS, we have a very early demonstration that you can do this well. And we're hoping for a successful outcome with our first collaboration that will make that something that we can hopefully use to influence the direction of research that's funded by the NIH and that's conducted at the CDC as part of our advocacy platform and as much as our research platform. And I just have shown here a one-page announcement about the biobank that we used when we announced the launch of the biobank to our readers of our paper publication. We also did so online, but it's just a little easier to show a print piece than multiple pages on a website. So I think that is my last slide. Yep.
Thank you, Kim. We really appreciate you telling us your story of how CFIDS began their biobank and all that you've accomplished in the past year. And as Kim said, CFIDS is a member of Genetic Alliance Biobank and is part of our network. But what I'd like to do now is to turn it over to Megan O'Boyle, who is the chair of the research committee of the Phelan-McDermott Syndrome Foundation. And I met Megan in January when she attended our registry and biorepository boot camp that we had. And we realized at that point that we needed to start providing some tools for the advocacy community to understand and learn about biobanks and registries. So we conducted a meeting. And Megan came and she went on this whirlwind tour of due diligence over the next 6 to 9 months. And I know for their group that they've not completely finalized their biobanking decision. But the reason we asked Megan to present is she went through the due diligence and was really in the weeds of the process. And I think it's a great compliment to Kim's story. And I think that in these two presentations, you're going to have many questions for our fantastic presenters. So Megan, please take it away.
Great. Good morning, everybody. And Liz, one correction. Thankfully, I am the registry and repository subcommittee, but there is another person, Geraldine Fliss, who heads up our research, and this is just one piece that I got involved in. And actually, I was not involved until January of this past year, so it's been less than a year. Just a little background on what Phelan-McDermott syndrome is. It is a genetic and seemingly rare syndrome. It was first identified in 1988. One case was reported, and by 1992, there was a small number of families who met over the phone, and in 1998, I believe, was the first in-person meeting of a few more families. And now we meet every 2 years. I think there were over 100 families that met last month. And as of this call, there are less than 600 identified cases of Phelan-McDermott syndrome in the world. So it is in contrast to what Kim just described for chronic fatigue syndrome. The Phelan-McDermid syndrome is diagnosed when a patient has been identified with a deletion or a mutation of their 22nd chromosome in a particular area. It was first described as 22q13. And it presents a spectrum of ways, and not everybody presents the same way, but it is considered a genetic cause of autism spectrum disorder. And I am a parent of a 10-year-old girl that has Phelan-McDermid syndrome. And hopefully we'll be able to communicate to you what I have done in the last few months. So as Liz said, in January I attended the Genetic Alliance Biobank and got my first introduction to what registries and repositories were. And I believe our group became aware of them back in October of '09, so less than a year ago, when we attended the ASHG meeting in Hawaii. So this has been a fairly fast process in terms of when we became aware of this and what we've accomplished. Now, in terms of why we need a registry and repository, we still need to characterize what the syndrome is, what the phenotype is. We need to collect data for a natural history. We are needing to verify the animal models that are coming up in research, and we need to identify the clinical endpoints. And we are hoping to better describe genotype and phenotype through studies and cohort development. This next slide just points out some of what we wanted to accomplish. We met in January of 2010 and established a scientific strategic plan, and at that time determined our goals for the research committee for Phelan-McDermid Syndrome Foundation. And over and over again, what we wanted to accomplish seemed to be reached with the establishment of a registry and repository. We believe that centralizing will increase researchers' interest and increase the presence of Phelan-McDermid syndrome in scientific publications. We also want to support a wide range of PMS-related research, not just what the researchers are finding grants to work on, but areas of research that would address all the medical issues that our children experience. We would want to support natural studies and develop useful research cohorts. We also, as I said before, need a better understanding of what the PMS phenotype is. And this really would improve clinical care and draw attention to currently unaddressed issues. And right now, for a family to receive genetic test results and be told that their child has Phelan-McDermid syndrome, almost never does their pediatrician, neurologist, or geneticist able to tell them much about the syndrome without having to go to our website. And we'd like to get much better information out to the clinical world and have that experience reversed. We also want to engage families in participation, and this will give us an increased amount of data on our families. As I said, there's less than 600 identified. And as a parent, I can tell you that if I am participating in research, I feel that I am doing something for my child, and this is huge. And ultimately, we want to facilitate clinical trials. And I think in the past, developing or collecting data and samples tended to be done much later in the game. And pharmaceutical companies were waiting on groups like ours to gather samples so they could get on with their studies. And we feel that doing the collection of data and samples at this stage makes a lot more sense. So our process started really in February, and it began with some web searches of who does this. I had some wrong assumptions that a lot of people did this, and that we were just going to be following along what other rare disease and genetic diseases did. And with a few phone calls, I'd find out who was in the business and talk to a few disease groups and find out what we needed to do. And as it turned out, I was completely wrong. There were not a lot of people doing this for groups our size for the purpose for which we need it. And even more shocking to me, there were not a lot of disease groups that had been around longer than us and had bigger budgets than us that had gone down this path. So I had to dig a little deeper than I expected, and we got a tremendous amount of information from that boot camp in January from Genetic Alliance. And in their resource repository, they have sets of questions that I pulled out and sent to the 4 or 5 vendors that I had identified for each of the registry and repository. Another assumption I had made was that all vendors would be able to handle both pieces, both the registry data and the repository of samples. And again, this was a misconception. So I simultaneously attended symposiums on the subject that mostly occurred at or near the NIH. I contacted about 30 different rare and genetic diseases and asked them what they were doing and how they were going about it. And I sent out these questions that I had gotten from the boot camp to the vendors. I also contacted current clients of the vendors that we had identified and asked them if they were happy with how things were going and what they had gained from it, and this was very valuable. And in some cases, because I live on the East Coast, I was able to actually visit the repositories and vendors, and that was very helpful. And during this process, our committee had designed a criteria that we thought the vendors needed to meet. So I took all this information that was coming in and compared it to everybody. And this next slide will indicate what we felt was important. The technology, the quality, the customer support, the customer usability, and compliance. And basically, when it came down to it, there were vendors that could do everything, and there were vendors that could do one thing really well but not help us in other areas. And some of the examples were, there were some vendors that were fabulous at software, but did not know anything about disease groups, and consequently could not do much in terms of customer support, or as I say, hand-holding. Especially, this is essential with the groups that's our size and does not have a paid staff of medical and scientific directors that can lead us in this area. We were particularly interested in having help with consenting donors properly and creating the best possible questionnaire that would provide the best data for the research community. And we knew that we would want assistance with IRB approval. This is something Kim also highlighted. There were groups that were happy to hold our hand but hadn't got to the technology standards that we needed yet. We're still working off of paper questionnaires, and we felt strongly that starting off with a web-based program really made the most sense. And there were vendors that met all of our needs beautifully and were just not anywhere near our budget. Our next step was to communicate the importance of why we needed this registry and repository to our board. And I don't have a slide for this, but it was a fairly easy point to make. If we collect better data from more patients and we get more biosamples from more families, and we own all of our data and our samples, then we can make them available to all the researchers around the world that we want to make them available to. And data will be shared, and samples can be shared, and that science is sure to expand while we're saving researchers time, money, and effort. And most importantly for us as a patient advocacy group, we wanted to save our families the trouble of traveling to different researchers and answering multiple surveys and having their samples harvested repeatedly. So when you boil it down to that, it's not a hard argument to make to a disease group board of directors that this is something that we need to do, that it's not something that should be left up to different silos around the world to collect data and samples and to keep it to themselves. On this slide, I just have a few of our things I've already covered. There were less vendors in the business than we expected. There were less disease groups that had done this already. There were some that had done their own, and we decided not to go down that path for various reasons. We felt for our group it was important to retain the ownership and control of our data and samples. And once this decision was taken, that led us in a very specific path. We also determined that we would rather have a vendor who did this for a living manage this than have our volunteers be responsible for creating and managing a registry. And we also understand that the input of the research community is vital to knowing what to collect and what to ask. I thought to myself what would be helpful to other disease groups of different sizes and have different purposes and maybe are at different stages. And this last slide is just some things to consider. Whether a group is ready for this undertaking, whether they find ownership of data and samples to be important, that the understanding of what the importance of patient consent are, and that it's been well researched in terms of what needs to be included in the consent, and ultimately who the patient group's data and samples should be given out to or sold to or shared with. So these are all things I think that every group needs to consider when they're doing this. And Liz, I think I covered most of it.
Great. Thank you, Megan. And so we will open this up for questions in just a few moments, but I actually wanted to share a few slides of my own. So Tam, if you could put my slides up, that would be great. And to just tell you a little bit about our resources and where you can find them, and then we've saved quite a lot of time for questions. So next slide, please. So at Genetic Alliance Biobank, we are a co-op model, and we offer registry and biorepository solutions for nonprofit organizations, and we share the resources. And we've developed a schematic that just sort of explains how the process works, but really what we've enabled is the infrastructure for advocacy organizations to manage registries and biorepositories, where they are consenting participants. They are having kits sent from a vendor to the participants, and then they're also contacting participants directly to complete clinical information. So we've really developed a system that can be used anywhere. It can be used with people within the United States, with people who are international, and it can be administered from anywhere, because we know that many of our advocacy organizations, our member organizations, they're in different places, and within each organization they often have people who are in multiple locations. Next slide, please. So we have developed this system, but we realized that there were a lot of people out there who were thinking about this. And we've done this and we put this together, and we really wanted to help provide some of our thinking and how we got to here. So on our website, we've continued to add a lot of resources on the biobank.org website, and we have a tools and training tab to tell you briefly about some of our resources. And if there's anything you'd like to participate in, please send me an email. My email is on the last slide. We've developed the Biobank Ambassadors Program, which is a forum to connect organizations who have established registries and biobanks with those who are thinking about it. We met at the Genetic Alliance Conference, and we had a breakfast together to really build the connections between the people who are doing this, because there are many shared experiences, and I think that people can really learn from each other. We've developed tools and training materials. We had our Biobank Boot Camp in January 2010, and you can see all of our Boot Camp materials as well as the downloadable templates that I'll discuss in a little more detail, and Megan mentioned. We have a listserv discussion for people to connect about biobank questions, and we've also developed a video where Sharon Terry tells the story of the PXP Biobank, which is a fantastic tool to really show why advocacy groups should be involved in these kinds of initiatives. And I think it's a great training tool to help explain to someone who might be skeptical. And then we also post our webinars here, and as well as on the Genetic Alliance site, and this one will come up shortly afterwards. And so what we've done, oh, I didn't realize this slide was animated. So we have 3 pieces that we created for Genetic Alliance. We have a step-by-step startup guide. This is for the boot camp, and the link below will take you to the boot camp agenda. We also have a vendor assessment tool, and then another tool to assess organizational readiness, which is really all about capacity and your board and your finances and sort of the infrastructure and legal aspects that need to be considered, whereas the startup guide is more about the science. Next slide, please. And so in the startup guide, we cover the areas of what will you collect, how will you collect it, and what else do you need. And so they really ask some targeted questions to help to figure out exactly what you want to do, because really the easy part is the logistics and the infrastructure. The hard part is deciding what really needs to be done for each condition, because each group and each condition is different and your communities are different. And that's the piece that only you know. And that's the piece where it might seem that the science is the hard part, but really the science has been done. And there are models that you can plug into for the science. It's the customization for your condition that's a little bit more challenging. And these questions really help you drill down to what's most important for you. Things about how to collect it in terms of regulatory compliance, operations and procedures, and both registry and biorepository considerations. Governance is a huge, huge issue, and I think it's the one thing I could emphasize. It's very, very important to think about who owns samples, who has access to samples, who determines who has access, and those kinds of things are covered in this section. And then there's some other information about other things like public relations and materials, and you saw that Kim had developed some really nice materials to announce the Solve CFS Biobank to their community, and then the process for making samples available to researchers. Next slide, please. And then with the vendor assessment, there is a summary that asks general questions, and there's some really specific questions on an Excel worksheet that talk about the data components as well as the biorepository components, even to the specifics of the types of samples and how they're handled. And again, these can be downloaded from our resource repository. And this has a lot of detail. Some of you might not be, some will be interested in the very detailed and others won't, but it is a pretty comprehensive resource. And then next slide, please. And then finally, the organizational readiness checklist, which talks about goals, support of key stakeholders. I mean, it's obviously essential that the board and the community are behind an initiative of this magnitude. Questions about funding and how to think long-term to make sure it's sustainable. And then just some operational procedures, because these projects tend to be a little different than some of the other projects that are typically conducted by advocacy organizations. They absolutely can be done, and I think advocacy organizations are well poised to do this. But there are just some things that I think it's good to think about. So we developed these to really help you, and we hope you'll use them. And if you have any questions or if you'd like to talk about your specific situation, please get in touch with me. And what I'd like to do now, since we have about 10 minutes left, is to start the question and answer session. So please start sending in your questions. And while I'm reading through the questions, I wanted to ask a question to Kim to get us started. And Kim, I thought Megan did a really nice job at the end saying what was important and kind of the advice she would give to others thinking about starting this. Now that you're in the midst of a pretty successful biobank, what advice or pearls of wisdom would you give to other organizations who are considering this?
It is definitely doable, and I think organizations of very different sizes and capacities and resources all have an opportunity to undertake a really exciting and dynamic project like this. But my sort of words of wisdom would be: don't underestimate, don't give short shrift to how much time and intensive effort it will require. This isn't sort of something that can happen in the margins of everything else you do. It really does take a dedicated group of people, whether they're staff, paid staff members, or volunteers. Really varies by organization, but we've just found that we're sort of biobank central for the last few months because we needed to really do an accelerated startup and to take advantage of enormous research opportunities that were very time sensitive. And so we, had to make some prioritization decisions about how other projects would sort of need to be scaled back or put on a different timeline in order to accommodate this because, we're all, while the Genetic Alliance Biobank framework and materials and resources are rich and thorough and extremely well accessible, it still is kind of a learning process day by day, and individuals' questions about things take time to adequately address, and we're just trying to do this in the real spirit of how this biobank came to pass, so that we have the kind of asset as it builds that we really know that it can be. And that has just taken, quite a bit of time, and I think we're refining every day. It's a process of sort of quality improvement and learning, about what we could do better the next time. We send out issue sort of another call for participants or engage with a researcher about discussions about samples and constitute a medical advisory committee to look over proposals. Each step we're just trying to make it as both efficient but most of all effective as possible.
Great, thank you. So Kim, do you charge the researchers for access to the data or samples?
We do. We do. And we've kind of come up with two different costs, and it actually depends. The first study that we are in the midst of right now is with an industry group, and they had very specific needs as far as the way that the cells were collected, the blood was collected, and the cells were processed. And they were willing to support that in terms of the fee that they paid per sample. But some of our collaborations that we're in the midst of developing, one is with another large national nonprofit, one is with a government agency, and then there are several academic institutions. So we're trying to be quite flexible, and for some of these have a very strong interest in the outcome and may kind of partner on the costs, and for others, it's a way to recoup some of the organization's operating costs that have gone into building the biobank through selling, the samples at more of a fair market value, if you will. So it's customizable from that sense as well.
Great. And I know that for other groups, some groups, if they are working to encourage research, they may have a nominal cost and require shipping. Some groups do not. They make their samples available to members of the research consortia, and I know that there are other models where research consortium members will write into their NIH grants costs for accessing samples. So there are ways to provide some costs back to help cover the cost. I mean, I don't, from my experience, I don't see these as money-making endeavors, but I think that you can cover some costs or all costs depending on how you do this. So I'm going to take the next question about issues that come up about collecting data internationally. And actually, collecting data internationally is quite easy, especially with web-based systems. I mean, really, it just depends how your IRB is written and that you, in your inclusion criteria, that it can be people in the U.S. or internationally. I think that in samples can be done as well. We just need to, samples can be a little bit more challenging because of customs regulations, and some things that are just really out of people's control, and this would occur for no matter what biorepository you were using. We just did a collection in Libya, and we used a different tube type that was more stable, where the blood could stay in the tube longer, because different tube types have different sort of shelf lives for how long they can be filled with blood before they're processed. So I don't think international is a hurdle. It's a little bit more complex, but it absolutely can be done. So this is a question about back to charging. How do families feel about the organizations charging for samples and data? Is that built into the consent? And it's not built into the consent. So Kim, how have you communicated to your participants, or that the pricing structure, is that more of an internal decision?
It's, we communicate that there is no cost to the biobank participant to be part of the biobank. You know, we don't charge a fee for them to be part of the registry or the biobank. Sometimes there is a cost for them to have the blood drawn for just the phlebotomy costs, and we've kind of created a sheet to help them minimize that by giving them, you know, names of labs or places to try within their community where they might waive the phlebotomy cost because this is connected to a research study. And then we do let them know that the samples will be made available only to researchers for projects that have been approved by a medical research advisory committee, and that there may be some costs associated with the acquisition of samples by the researchers. But we don't get specific in terms of how much, and that's essentially to offset the direct costs of the tubes and the biobanking and the shipping and the cell processing and everything that sort of happens on the back end that they aren't a part of. But we don't get specific in that detail because, as I said earlier, it does have some variability depending on what type of study it is.
Great. And Megan, this one is for you. So would you start with collecting data and samples and actually just starting the registry, or would you start with public awareness in your community for people to donate to future research? So would you start with a biobank, or would you just start with the awareness campaign?
We would start with the biobank, but we would obviously have to educate our families as to why we were collecting and why this is different than other times that researchers have reached out to families. And we'll need to continue to educate our families because we'll constantly be getting new families for starters, but just to make sure they understand the importance of what we're doing and why we're doing it, and that it's ultimately to reach more researchers and to expedite everything through transition.
Great. And I know that we have come to the end of our time. I wanted to let I wanted to thank our participants for joining us, and I'd really like to thank our presenters for telling their stories. I know that not everyone's questions are answered, but we will get those answers to you. And I also wanted to let you know that the samples are, the presentations are in source repository, so if you go to our website, to the webinar page, you'll be able to link to that. And I hope this was helpful, and as you're thinking about these things, if there are other things you are interested in, developing a multi-part webinar series about this. We'll be covering governance issues later in the fall. We'll be covering informed consent and IRBs, just to name a few. And if there are other things that you'd really like to be covered in this format, please let us know. And again, thank you so much for joining us. We really appreciate it, and we hope you have a wonderful day. Thank you.